Hi all, Dr. Clark here again. Today I'm just going to give kind of a brief overview of the phyla Porifera. Porifera is the phylum with the common name sponges. Sponges, I guess you could say, at least tentatively, right now as I'm delivering this, are the most primitive animals we have in the animal kingdom. And you'll see a lot of features in Porifera that are very similar to protozoa or the protista kingdom. Porifera, however, have some animal-like features, and so it's kind of this transition phase from single-celled aquatic organisms that may be free-swimming, may not, in the form of protista, to Porifera, where the cells are starting to come together and starting to work in collaboration with each other, not to the point of being considered tissue, but enough that you can have a whole organism with a few different types of cells. Okay, so let's progress through Porifera. I'm going to give you some information about the general biology of Porifera, and then we're going to talk about a few classes in Porifera. We'll talk about reproduction, etc. Okay, so this is a calcium carbonate sponge belonging to the class Calicarius, and that just means that the spiracles, and we'll talk about this a little more, are made of calcium carbonate. Okay? And we'll look at these different features, but the osculum, you can see these openings, you can see at kind of the dorsal or the top portion of the organism. Okay, we'll come back to this and we'll look at anatomy in a second too. So first I want to talk about Porifera from an evolutionary perspective, along with some other groups that are kind of linked in the same grouping as Porifera. So first of all, Porifera, they have no tissues. Okay, so the cells that they do have are independent, and so they have independent labor. Now that doesn't mean that there is not a little bit of coordination going on between cell types or even within the same cell type. There is, but because the cells are not linked and they don't work as a concisive unit, it is not considered tissue. Okay? The interesting piece, or the lack of information that we possess when it comes to Porifera and from an evolutionary perspective, is that they have their own ancestral connection. They have their own ancestor, and it's independent of other animal ancestors. Right? Those other animal ancestors would be cnidarians and ctenophoras and things like that. So we're interested maybe in, well, how did they develop? What protista, what protozoan gave rise to Porifera? We have fossil evidence of ancient sponges. Okay? The problem that we have when examining fossil evidence is that it's hard to see at the cellular level, are the cells that we currently see today, these independent cells, are they the same cells that we saw, millions of years ago, hundreds of millions of years ago? Okay. Right now, we believe that the precursor to Porifera, or the protista that gave rise to Porifera, to the sponges, is a group of protists called the cyanoflagellates. And I'll show you why that is in a little bit when we start looking at the anatomy, the inner workings of Porifera. There are two other groups that kind of get lumped into the primitive animals, and that would be cnidarians. Okay, so these are jellies and corals and things like that, and ctenophora, which they don't really have a common name, they're just called the ctenophoras. which are kind of like jellies, and you'll see that superficially they look just like jellies, except for they have some other unique features. When we start looking at them, we now see tissue level organization. In Cnidaria, they're diploblastic, so they have 2 layers of tissue. And in Ctenophora, they're nearly triploblastic. Okay, so their mesoglia cells in Ctenophora have they have developed muscle cells inside their mesoglia. Now, it's kind of like Porifera in that you have cells in that region, but they have independent labor. So Ctenophora is still lumped as being diploblastic, but on the verge of triploblastic. We'll come back to these. We'll talk more about these in further lectures. Again, both Cnidaria and Ctenophora have independent origins of other animal ancestors. They're independent of Porifera. So this is a polyphyletic group, not the monophyletic group that we're after. And again, probably the result of channel flagellate protista giving rise to them. Now, a different group of channel flagellate protista, different ancestral line, possibly. It could just be that Porifera, Cnidaria, and Ctenophora all share a common ancestor. We just haven't found it yet. We haven't dug deep enough. We don't have enough information to find that similar or that channel flagellate that might have gave rise to this evolutionary line. So we're pretty confident in that the origin of multicellularity is at least 800 million years old. Now, recent findings suggest that it's quite possible that multicellularity is up to even 1.5 billion years old. And that's based on some evidence that was just found in the fall of 2018, in that there are tracks of organisms that can move, and seemingly fossilized tracks of organisms that can move that are dating all the way to 1.5 billion years ago, and they seem to be multicellular, at least based on the evidence of the tracks and the remains of the organism. So, if we start looking at the evolutionary relationship, when we start looking at Animalia, the kingdom, so we move from Protista, and if you remember what I said, this is also a polyphyletic group. There's quite a few different protists. Okay, they all have a single ancestral line. Could be the result of not enough information. Could be the result that these shouldn't be lumped together. Whatever it is, this is polyphyletic. Same deal here. Porifera, Cnidaria, Ctenophora all have single ancestral lines. It could be that these should not be lumped together, and they should be separated in their own superphyla. Or it could be that we just haven't found that common ancestor that connects all 3 of these. Okay? But we're going to concentrate on Porifera for this portion of the lecture. In order to examine that connection, that evolutionary connection to channel flagellates, or that protista connection, there are 2 hypotheses that are currently out there. One that has a fair amount of support, and then one that is favored by a few individuals. Okay, and the first one is that the organisms developed, or multicellularity developed, from the colonial hypothesis. In other words, you had these potentially channel flagellate protista that clumped together. Okay? We still see this today in environments where conditions change. You'll get clumping of protista. Okay? And so they would have clumped together. By clumping together, they could work as a colonial protist. We've seen this. We have examples of colonial protista today. Then from there, you could get 2 varieties. of colonial protista lumping together, at least two, okay, and creating a kind of a radial ancestral. And so two-layered, different kind of protista on the center and different on the outside, okay, and that would have evolved into radial animals. And you'll see that Porifera is nothing more than this picture right here. So that's one hypothesis, that radial animals evolved first and bilateral animals evolved later off of the radial animals. The second hypothesis is the synethal hypothesis, in that we had one large protista that then divided and developed boundaries within itself. Some kind of cell membranes were formed to divide itself. That division then became bilateral division, in which the organism was split in two, and bilateral animals came first and radial animals came after the bilateral animals. So two general hypotheses. The colonial hypothesis has probably the most evidence for it. Okay? But it's difficult to figure this out because some organisms are, and primitive organisms are bilateral animals. Okay? And for a long time we thought bilateral animals came first and then radials came after. But it might be that radials came first and bilaterals. So neither hypothesis has been developed into a theory. So they're both hypotheses out there. They're both been tested. Sometimes they're rejected, sometimes they're not rejected. It's really a matter of, we just don't have enough evidence at this point in time to decide which one of these two hypotheses is more right, or at least supported more by scientific evidence. Okay? But we'll talk about this a little bit more as we progress. I definitely lean to the colonial protist hypothesis because there's just more evidence for it, and when we start looking at Porifera, that seems to be what's going on in Porifera. Alright, so let's talk about the phylum Porifera. First of all, there is no symmetry in Porifera. So regardless of whether it is radial symmetry or bilateral symmetry is the first organism, Porifera themselves are not symmetrical. Now, as they develop, some of them will have a superficial radial symmetry, meaning that it looks radial. Okay? But often there's still no symmetry. When you divide it, it's not divided equally, either on a central axis or on a bilateral axis. It's not divided equally. There are three cell types. Remember, there's no tissue, no symmetry, okay? But they have three different cell types. They have pinocytes, mesenchyme cells, and chanocytes. Okay? Now, they are all have kind of a specialization. They have different functions, and we'll talk about them in a little bit. Majority of Porifera are set up with kind of a central cavity in which they're going to filter feed out of that cavity with their specialized cells, particularly chanocytes. Okay? Now, off that central cavity, they may have a whole series of branching chambers and things like that. And we'll talk about the different types of kind of internal structure in Porifera. And this determines a lot of things like, well, how big the organism can get, okay? How efficient it is in feeding and reproduction and things like that. Depends on how many chambers and how much surface area they have. Because these are filter feeders, so they're sucking in water and removing food out of the water column. Okay? And the more surface area you have, the better that you are at doing that filter feeding process. Okay, so again, just to reiterate, there are no tissues, even though they have cells, and the cells sometimes coordinate with each other. They have no tissues, and if they have no tissues, they clearly have no organs. Alright, three main classes. Now there is some fluctuation in this. So there used to be a class called sclerospongia, okay, that has been moved into demospongia. There is a new proposed class, okay, that it would add a fourth class to Porifera. Now, it's not widely recognized yet, but it might be a legit class. So again, like I said before, when it comes to animal biology, I'm going to give you the information that we have currently, today, that's been confirmed. But that doesn't mean in 10 years that lots of things don't change. In 10 years, it could be that Porifera is its own group, and it's not lumped with cnidarians and ctenophores. And maybe it has more than 3 classes. Maybe it has 4 or 5 classes of organisms. And so it really just depends. A lot of the early primitive animals that we're going to talk about, this classification or their classification will change a lot in the near future, mainly because they haven't been studied to the degree that higher order organisms or more derived organisms have, like mammals and avians or birds and reptiles and things like that. Those organisms have been studied a lot, and the classification has been worked out pretty well. Classification of primitive organisms, not so much. But at any rate, 3 classes that I want you to know. The first class, and these classes are really divided up by what is their body made of, or what are the spiracles, which are kind of they're structures that allow for them to hold shape. Okay, what are they made out of? Okay, and that really determines what organisms going into what class. So Calicaria is made out of, the spiracles are made out of calcium carbonate. Right? So they're kind of hard spiracles. These organisms are normally called calcareous sponges. They're not flexible. They're not. They're more rigid. Right? Not as rigid as Hextanilidae. Okay? The hex, or what sometimes is called the glass sponges, their spiracles are made out of silica. Okay? And they're really fine, and like the name glass sponges, they do break, and they shatter, and are fairly fragile in that sense. But again, no flexibility really to the outer surfaces. They're kind of rigid. So both Calicarius and Hextanilidae, okay, are that kind of rigidity, have that rigidity. Okay, Demospongia is what most people think of when they think of sponges. It has that flexibility. Okay, most people have used like true bath sponges, or they've used them maybe to paint walls and make designs, this kind of stuff. Those sponges are, once living organisms, and what's left is the external skeleton, right, which is made of spongin, okay, typically made out of spongin. Okay? And that's where they get that flexibility of the organism. Okay, so 3 main classes, really divided by what is that material that's going to hold their shape made out of, either calcium carbonate, silica, or spongin. Now, Demospongia can also have silica spiracles. It's true. Okay, so it's not all about just what you're made out of, but for the most part, if you're made, if you have silica spiracles, then you belong to Hextanilidae. Okay, so back to those cell types. The pinocytes, okay, are the outer surface of the organism. Okay. Some have the ability to contract, close off pores, okay, and or open up pores, okay? And really they're associated a lot with that filter feeding mechanism. So closing off the osculum or the osteolium, okay, allowing for water to flow in, shutting it off so the water can't flow in, these kind of things. That's really the idea, the premise behind the pinocytes. Mesohall is the jelly-like substance that you find inside the outer surface, between the outer surface, which we call the ectodermis, and the inner surface, which we call the endodermis. You're going to have kind of a jelly-like middle layer. Okay? Now, the mesohall, or the mesoglia, okay, is devoid of cells, typically, in sponges. So there's no cells. And if there are cells, the cells are either coming from the ectodermis or the endodermis, but they're not mesoderm cells. So they're not derived from inside the mesoglia or the mesohall. And that plays an important role when we start talking about organisms that have two tissue types, etc. The mesenchyme cells, these are amoeboid cells. So they're similar to amoeba-like protista. Right? They're important in things like phagocytizing material, making food vacuoles, things like that. Reproduction. Sometimes, depending on the species and the class of sponge that, or the class in which the sponge belongs to, they can help with excreting calcium carbonate to create that rigidity within. Okay. And the channel sites, these guys are very important for creating the water current. Okay, they have a flagellum which they beat, and this is when I was talking about having some coordination. Channel sites will have, in some cases, will have coordination. So they'll all beat their flagellum in a single direction to create a water current. So they're not fighting against each other to create that kind of current. That current will bring in food particles into the osculum, okay, or through the osculum, and allow for them to filter feed. Now, the other piece of the channel site is a lot of them will have a ring around them, okay, and so they have a ring that's close to the flagellum, which allows them to pick up food particles and create food vacuoles from that. And so we call that the collar of the organism, or of the cell. So they have channel sites that are collared, and they'll pick up food, create a food vacuole. Sometimes that collar goes away for reproduction. We'll talk about that in a second. Okay. Their skeleton, like we talked about before, can have spiracles. Okay, so these are rigid features. Okay, or they can have spongin, which spongin is more of a protein base, a lot more flexibility. Okay, and that's what allows sponges to kind of flex and then go back to the original shape. All right, so let's look at some of this morphology that we were just talking about. Okay, the osculum is the main opening of the organism. Now, it's showing that, you know, you only have one direction. This really depends on the organism. Water can flow in through the osculum also. These guys are filter feeders, so they can bring water in through the osculum and back out of the osculum. Okay, the ostium, or ostium, okay, are the pores that enter, okay, into this cavity, okay, from the side walls of the organism. Okay, that's where your like porocytes would be, which would allow for that material to come in. The pinocytes are going to control that that opening typically. Okay. The amoeboid cells will be inside the ectodermis. Okay. And again, this is depending on the species. This could be reproduction, but this is also where excretory material to make spiracles are going to come from. The channel sites are the main players because they're involved directly with feeding. They're involved directly with creating that water current, so water is going to come in through the beating of the flagellum of the channel sites, which occur inside the central cavity here. And again, they have these collars. Typically, they have these collars of thin membrane, okay, which will trap food, and then they can phagocytize that food and make a food vacuole, which then inside the channel site, they would have lysosomes, which would then break down and digest that food, and they have a nucleus and a typical system for breaking down food. This should remind you of a protozoan, of Protista, in the sense that they're going to trap food, bring it in, consume it, okay, and that's how they're going to, energize individual cells. So each one of these cells, even though they might be in real close proximity to each other, they act individually. Right? So yes, this is a solid organism, but there are no tissues. There's just individual cells that act from an individual level. Okay? Again, there is some coordination depending on the organism. So all the flagellums could be beating in a single direction, or this side could be beating in one direction, this side in another, so you can get a water current going. But nonetheless, it's not enough to call it tissue. Here's an example of some spiracles. Okay, so this is a calcium carbonate spiracle. Okay, that would be embedded in between kind of the ectodermis and endodermis in that mesohall, mesoglia system to provide that rigidity of the organism. Again, like I talked about before, there are different body forms within the different classes of porifera. Okay? So you can have ascion, scion, or lucyon. Okay? And this really just is determined by basically how much surface area does the organism have. Okay? Is it just one central cavity, okay, with a single osculum and ostia that feed into that osculum? If that's the case, then it's ascion. Okay? Is it a single cavity but has multiple folds within it, which will increase surface area, allows the organism to get larger? Okay, they might have different types of pores that enter. Okay, and they could have more pinocytes that would control those pores, etc. Okay, then it's scion. Is it much more complex than that, where you have these chambers that would then allow for different features to occur at different times? So you could have feeding over here, you could have reproduction in this region. Okay, these would be what we call channel site chambers, okay, that have the ability to increase the surface area much larger, okay, and therefore these organisms are going to be the largest. The lucyon organisms are going to be the largest sponges. Okay, so when it comes down to body form, especially in porifera, it's all about surface area. The more folds the organism has, the more ability it has to filter out more nutrients. It's more efficient at filtering out nutrients. It can conduct, feeding and reproduction at the same time. Okay? And so it's just more efficient. That efficiency increases or allows for the organism to increase the size of the organism and the amount of water in which it can filter at any given time. Right? All right, so filter feeding. What are they eating? Mainly eating things like bacteria, algae, other protists. I shouldn't say other protists. Okay, protists that would be, smaller than the channel sites that they have within the water column. Okay, so again, they're not chewing things up. Anything that's too large to be trapped as a food particle in the collar of the channel site would just be pushed out of the system again. Okay, so the channel sites are going to trap it, incorporate it into food vacuole, and then, like I said before, Digestion is going to occur because, enzymes, digestive enzymes from lysosomes, okay, or even some pH changes and things like that with inside the cell is going to help break down, denature any proteins and things like that, and allow for them to, break them down into their smaller portions or smaller parts. The waste is diffused outside of the cell, so it goes typically through from the channel sites, okay, back into the water column within the central chamber, or if it's closer to the external environment, okay, it would go out. The pianocytes are out, the ectodermis, that outside layer, out into the water column. Again, like I said before, there is some coordination. So it's moving from being single cells to a collaboration of multi-cells, even with some coordination. Not enough for it to be considered tissue, okay? But on its way to evolving into tissue. Reproduction, these guys are monoecious. If you remember, monoecious means that they have both male and female reproductive parts. Now, for the most part, monoecious organisms are not self-fertilizing. So the sperm from the same individual is not fertilizing the egg. Often, the protein coating on the sperm won't even be recognized by their own eggs, so they won't have the right bindin, which is a protein coating on the, or they won't have the right lysine, which allows for the sperm to enter the egg. Okay, but nonetheless, porifera are monoecious, in which they are both male and female. Okay? The channel sites, okay, are the main reproductive parts, but sometimes, like I said before, the amoeboid cells can go through reproduction. Channel sites will lose their collars, okay, and their flagella, and then they just undergo meiosis. Okay, so they divide, and then it will produce sperm-like or egg-like gametes. Fertilization is external, so the sperm is released to go find eggs, the eggs are released to go find sperm, etc. in the water column. Fertilization occurs, zygotes form, and then that larvae is going to be planktonic, okay? Which means it's going to be moving throughout the water column, okay, as the form of what we call plankton. Now, reproduction can also occur asexually. So you can get the formation of gemmules, which is basically small branches or buds of the organism can break off and form a new organism. As long as there's the right material there, as long as you have the 3 cell types and you have some ability to get established in a new region, you can have this asexual reproduction. So, it's quite common, for people, if they want, like, let's say you're have an aquarium and you want more sponges, to just cut them into pieces and they'll grow themselves. It's true, okay? As long as there's enough, or it's a big enough chunk, it'll regrow itself. Okay? Now, the other interesting piece about this reproduction is the term gemmules. Okay? This comes from, I'm not entirely sure if Darwin coined the term gemmules, but Darwin used the term gemmules to explain sexual reproduction and inheritance of traits from their parents, from, male and female, of the parent of that offspring. We're talking 1850s, even before that, okay? We had no clue that there was such thing as DNA, chromosomes, or anything like that, so we didn't know how things were inherited. Now, Mendel was working on this in the 1850s, but Darwin and Mendel didn't know each other, so Darwin came up with his own idea of how this worked, and he called it gemmules. In that little teeny pieces of all the organism, okay, Okay? So if we're talking about humans, a little teeny piece of our liver, kidney, okay, our stomach, our heart, lungs, everything would break off, small pieces would break off, and they would float down, okay, into the gonad region, get incorporated into sperm. That sperm would pass it on to an egg, okay, and the egg would have the exact same thing occur, get incorporated into the egg, and then an organism is a combination of the egg and the sperm's gemmules, little teeny pieces of all the body functions. Okay? Now, we know that Darwin was wrong, but that was his hypothesis on how organisms became, basically half the dad and half the mom. Okay? We do know that in asexual reproduction, that's exactly what's kind of going on with sponges. They do form gemmules. So that chunk that breaks off can get established, and it would have enough of the three different cells to establish itself and become basically a clone of the original of the parent. Okay? Now, it can occur in some marine systems. It occurs often in freshwater systems. You might expect this given that the planktonic larvae is sometimes difficult to have it occur in a freshwater system. It's hard sometimes to have the sperm and egg encounter each other in a freshwater system. Now, I'm not saying that reproduction doesn't occur, sexual reproduction doesn't occur in freshwater systems, but it depends on the cycle. So if the lake's turning over and things like that, and you get a lot of movement, then a freshwater system would definitely be going through sexual reproduction. But if it's stagnant water, then a lot of asexual reproduction is probably occurring in those organisms if there's a lot of food and whatnot, and they're going to reproduce. Okay, so there are a couple different types of larvae and stages of larvae depending on the organism that we're talking about. We just talked about gemmules, this body, this chunk that breaks off, okay, and you can have these amoeboid cells and these spiracles, which allows for the organism to reestablish itself asexually. The two kind of planktonic larvae, okay, one being the pericummal larvae, which looks very similar to that colonial protista hypothesis, okay, would then form kind of a globular projection that would get established on some kind of substrate and then form the internal pores from the original colony of cells, okay? Or the amphiblastula larvae, which is going to have two different types of cells to begin with. You have an external cell which has no flagellae, and then you have the flagellum cells on the internal, which we call the chanocytes on the internal, which would eventually form. Okay? In both cases, this can occur. Okay? Well, in all three cases, the amphiblastula larvae is probably the most common. Okay? But nonetheless, all phases do exist. Okay? All right, so with that, that's kind of just in a nutshell, basic biology of porifera. Again, no tissue, 3 cell types, no symmetry, so they're asymmetrical. Okay, prehistoric, there's a little bit of fossil evidence, but they don't fossilize very well. So a lot of that information about, well, when did they evolve, that's lost, and it's very limited to the fossils that we do have. Okay. Next, what I'm going to talk about is Cnidaria, the jellies, and Ctenophora, okay, the jelly-like ancestors to other animalias. Okay, so we're moving from porifera to organisms that now have tissues. Okay, and we'll see kind of the connection between the groups and the differences that you would see in the two groups.